Good afternoon, everyone. I'm Jackie Spear. I'm one of the co-chairs of the Biomedical Research Caucus, and we're delighted that you are all here. You are hopefully going to take all you learned here today back to your members and regale them with how important it is for us to make sure that biomedical research is part of our platform as we move forward. I've got the great pleasure now of introducing our speaker. And how many of you have biotech companies in your district? Raise your hands high.
Really high.
All right. How many of you have academic institutions of higher learning that do a lot of science in your district? Okay. Interesting. So, in my district, which takes in part of San Francisco and San Mateo County, I have the distinction of being able to say that South San Francisco, in my district, is the birthplace of biotechnology. And it didn't happen by coincidence. It happened in part because of the confluence of UC Berkeley, UCSF, and Stanford University, all within 15 or 20 minutes of each other. And the fact that a UCSF professor named Herb Boyer sat down for a couple of beers with a venture capitalist named Bob Swanson in San Francisco on Union Street. If any of you have been there, it's a young singles hangout. And over the course of an afternoon, a 3-hour discussion, they shook hands and founded not just a company, but an industry. And that was the founding of Genentech, which is in South San Francisco with about 80 other companies. Genentech, you will hear from our guest speaker today, now has 10,000 employees. So it is critical to our future, and as you will hear today, that if we don't do something quickly in terms of restoring funding to NIH, we are going to very quickly see scientists in this country go to China, because that's where innovation will start to take place, and it will be too late to do anything about it. So let me introduce you to our keynoter, who is really remarkable, and overachiever doesn't begin to describe the genius of this individual. Jim Sabry is the Vice President of Genentech Partnering. He's actually from Canada, where he went to the University of Queen's University in Kingston, where he got his MD. He then went to Harvard Medical School, where he completed his residency in neurology. Then came to San Francisco and got his PhD in neuroscience and biochemistry at the University of California, San Francisco. And if that wasn't enough, he then went to Stanford and did his postdoc work in biochemistry at Stanford. He became the entrepreneur in residence at the Mayfield Fund in Menlo Park, California. And then he launched his very first company in 1998, Cytokinetics, and as CEO there, raised the company from infancy to more than 200 employees, which then the company went public in 2004. In 2008, he became president and CEO of Arete Therapeutics, a company focused on the development of therapeutics for diabetes. And in 2010, moved on to Genentech to take his current position. He's also a member of the board of directors of a nonprofit organization, Hope Lab, and is working on advancing high-tech solutions to certain diseases of youth and young adults. Put your hands together and welcome our keynote speaker, Dr. Sabry.
Thank you, Jackie. Thank you very much.
Actually, before we get started, I'd like to introduce this.
Hi. Thank you.
I guess you're going to be delayed from your keynote speaker for just a few minutes. I'm Steve Cohen, and I'm from Memphis, member of Congress from the 9th District, and a strong supporter of the National Institutes of Health. It is my opinion that the likelihood of any of you dying, or me for that matter, from some action taken by one of our political enemies, Korea, Iran, or any of those other places, are slim and none. Now, we got a good Defense Department, that's important, but nevertheless, I don't think that the idea of the Third World War is going to be that close. But I do think it's likely that you will meet your ultimate demise, as will I, from what are our true enemies. Cancer, heart disease, stroke, diabetes, Alzheimer's, illness. That's our real enemy, is illness. So our real Department of Defense is the National Institute of Health. And for each of us, don't get deluded. The National Department of Defense, for you, is the National Institute of Health. And for the Republicans who can't get it in their skulls that the sequester that takes $1.6 billion from that is wrong, because they're protecting the next generation, which most of y'all are part of, because y'all are going to have to pay that, y'all are also going to get the benefit of it. Because the cures, the treatments will affect you more likely than they'll affect me. So it's a fair deal, it's the right deal. Try to get your reps to vote to support the National Institute of Health for your sake. Thank you.
Well, I've never been introduced by two people from the Congress, and especially by my own Congresswoman, so thank you, Jackie, for having me. For the next 40 minutes or so, I'm going to tell you some stories about the pharmaceutical industry and the biotech industry from the perspective of Genentech. And I'm going to give you the key messages up front, so you'll know them. If you take away anything, take away that first slide. The rest of it is going to be commentary that gives rise to why these key messages are so important. And the fundamental message I'm going to try to convey to you is that we need the NIH in pharma. We actually don't benefit from the NIH. We can't get NIH grants at Genentech. The money doesn't come to us directly, but what comes to us is knowledge, basic knowledge of how cells work in a disease state, and how cells work where they're part of a complex human organism. And without that, our industry comes to a grinding halt in the United States. As Jackie pointed out, it's not that it'll come to a grinding halt, it will just move. We've already seen this happen in Europe, where science is basically moving from Europe to the United States. And with it, the pharmaceutical companies have moved their R&D centers from Europe to the United States. We now have 10,000 employees at Genentech. It's the largest site for Roche, which is a Basel-based company owned primarily by a Basel-based family, the Hoffman family. But they're not keeping their R&D in Switzerland. They moved it to San Francisco for a reason. Novartis did the same thing in moving its R&D center to Cambridge, Massachusetts. And they didn't go there because they liked America, although they probably do like America. They did it because the science is better and because they go where the science is. And if science doesn't stay in the United States, these companies will move their R&D centers to wherever the science is. The next place it's going to be, unless we make a difference here with regards to NIH funding, is China. And with it will go all the companies as well. The first point I'd like to make is from a technical viewpoint, there's never been a better time to be in the pharmaceutical or biotech industry. The amount we know about human disease right now dwarfs what we knew even 5 or 6 years ago. The drugs we're making are so much more effective than they ever were even a decade ago. I went to medical school 20-some years ago, and the drugs we had then don't hold a candle to what we have now. The drugs we have now are targeted to specific patients. They get at the essence of why the disease is occurring, and they effectively shut down those pathways. And what we're seeing when you do that, not surprisingly, is they're deeply effective. So for those of you that decide you don't want to work on the Hill, you can come and work with us in companies. There's never been a better time to be developing therapeutics for human disease. The demands for drugs are going up. They're going up because the societies are aging. And age is the single best predictor of pharmaceutical use. Evolution will keep you alive until you're 25 or 35, but after that, it's us. You have to take drugs after that. So there are many of you that are probably still in that evolutionary phase, but many of us are post-evolutionary. And so for us, we need drugs because our human bodies were not designed to live past 25 or 35 years of age. The second reason demand is going up is money. The richer you become in a country, the more per capita you have, more per capita GDP you have, the more you spend your discretionary income on health. So China, Brazil, Russia, these countries are getting richer, so people are living longer and they're spending more money. So if you're the CEO of a pharmaceutical company, you've got a great job because the demand for your products is going to continue to go up and up and up. Better than being, say, head of a newspaper or automobile industry where you're not sure the demand is going to go up. In this industry, it's going to go up. But there's a problem. Our productivity is going down. The productivity is going down so that right now, for every drug that we put into development in the industry, and this is true at Genentech as well, only 1 in 10 make it to the market. So 90% of the efforts, 90% of the billions of dollars we spend, and Roche spends more in R&D than any other company in the world. Not just any other pharma company, any other company. We spent $9.5 billion in R&D, mostly in clinical development, and we still have a success rate of 10%. That has to change. We know how to change it, though. There are 3 ways that we can change it. 1, we can work on antibodies or large proteins as drugs, and I'm going to come back to that. 2, we can use what are called companion diagnostics, where we subset the patients genetically so that we personalize the medicine for your genetics. And the third one, and the one I want to talk to you about here today, is understanding the mechanism of action. A deep knowledge of the science behind the drug is the largest predictor of success. So I'm going to tell you some science stories, but the fundamental message is an economic one. If we understand basic science, our drugs make it to market. If we don't understand basic science, our drugs don't make it to market. If our drugs don't make it to market, the company fails. And right now, the return on our investment is below our cost of capital. So for those in the audience that are economists, you know that industries can't stay in that place for very long. It's like running United Airlines, filling the flights are full and still losing money. It just does not work forever. And we're in that situation right now, and we've been there for about the last 3 or 4 years. So, aging. The purple lines are how quickly we're aging. By the time 2050 comes along, by the time for many of you, you're going to be in your mid to late adulthood, there will be more people on walkers with tennis balls in the bottom than there are in tricycles and in carriages. The country is getting older. And as I said, once you're past that evolutionary age, drugs are going to keep you alive. Very few people make it to 50 or 60 without having some sort of illness that's chronic and requires a pharmaceutical in order to continue to have a fruitful, productive, and happy life. We're getting richer. This is a graph of the log GDP per capita versus pharma spend per capita, and you can see it goes up and to the right with the United States peaking out here. This is a very rich country we live in, and we spend a lot of our money on healthcare. We spend a lot of our money on healthcare because it's discretionary. We want to. We have the opportunity to. And so as some of these countries that right now aren't quite as wealthy get wealthy, they're going to start doing the same thing. It's going to happen in China. It's already happening in Brazil. It's going to happen in these countries as they get richer. They're going to live longer, and they're going to choose to spend more of their money on healthcare. But this is the bad news. This is the productivity curve. This is a curve of the R&D spend in our industry in billions of dollars versus, with a 5-year lag so that the research investment can make it into the market, of the FDA-approved new medical entities. Now, it was a good year last year, but you can see that this has been relatively flat, and this is going up and up and up. Right now, we estimate at Genentech that the cost of getting a drug to market, including the failures, is somewhere around $3 billion for one product. So we have to recoup that R&D cost, plus the opportunity cost of not taking that money and using it somewhere else, somewhere in drug pricing. Now, rightly so, there's been an argument that drug prices can't go up much more than they are. So I think we're limited as to how much, as a society, we're willing to spend per pill or per injection. What needs to change is productivity. We need to get this number up from 1 in 10 to 2 in 10, 3 in 10, 4 in 10. And as I said, the major way we can do that is an understanding of basic science. So let's talk about this number. This is the most important statistic in our industry. It's called PTS, the probability of technical success. The probability that if you're building a drug and you put it in development, it makes it to market. 9 out of 10 of you will fail at this job. Even the best of you. Nobel laureates don't do any better than non-Nobel laureates. 9 out of 10 drugs don't make it to market. They fail somewhere in clinical development. And this is where they fail. This is a graph showing you the probability of success through the different phases of development. So 9% overall, as I said. For small molecules, these are molecules, and I'll show you a picture of one in a second, that can be taken as a pill. They're chemically synthesized. It's the classic drugs that you've always thought of. A drug like Lipitor, for instance, is a small molecule. It's 7%. Large molecules, a drug like Herceptin, if you have breast cancer, an injectable antibody against a cell surface marker for breast cancer, is more than twice as likely to work. So if you decided you were going to do the path that I did, start a biotech company, I'd recommend you work on biologics, work on antibodies. They're twice as likely to work. But when they fail in the various phases that we go through before we apply to the FDA for a new drug application. And these are the success rates from over 4,000 drugs in the last 6 years or so. And these data are pretty accurate to what goes on at Genentech right now. Most of them fail somewhere through clinical development, and they fail either because they're toxic, and we are not perfect at predicting human toxicity, or they fail because they don't work when you get them into the complex environment of the human, although they might have worked if you have them in the complex environment of a mouse or the complex environment of a cynomolgus monkey. But when you get them in humans, for some reason, they don't work. And again, the main reason they fail when you get them in the humans, if they've worked in animals, is because you don't understand how it worked. But it did work. And so you're excited. The drug works. It works in this animal model of Alzheimer's disease. I'm going to do a clinical trial. And the clinical trial turns out, as we have found out in the last 2 or 3 years from companies like Pfizer and Lilly, it doesn't work in humans. It doesn't mean the animal models were wrong. It just means they weren't predictive. And we do not have perfect predictive models for human disease yet. So this is the difference between a large and small molecule, just to make it apparent to you. This one is small. Oops, let me go back. This one is small. This one is large. There's the small one, and there's the large one. That's the difference. This one actually is a model of an antibody, a drug that's injected underneath your skin or into your veins because they can't be absorbed through the gut. These form the basis of cancer treatment these days and the basis of the drugs that Genentech has made for the past number of decades. And as Jackie pointed out, one of the things that started our whole industry were ways of synthesizing large molecules like this. The pharmaceutical industry, Merck, Pfizer, Novartis, have been playing around with small molecules for hundreds of years. These ones are chemically synthesized. They can be taken as a pill. They're so small that they can go right across your gut membrane and go into your system. And so when you take aspirin or Advil, it's a small molecule. You don't have to inject it. And it's these molecules that fail much more frequently than these molecules because they tend to go places. It still blows me away that you can inject a molecule like aspirin, which is about this size, and you don't fall apart. It goes everywhere in your body, but it binds probably to 1 or 2 major proteins and modifies the activity of those proteins, and that's why it's an anti-inflammatory. Blows me away that we can do that, but we can. But molecules that are so promiscuous in where they go and what they bind to are likely to be more toxic, and that's why these molecules fail. These big ones are designed to be highly specific where they bind to, and that's why those drugs are more likely to work. What I want to do now is tell you a couple of stories about science, about partnering, about Genentech, and about what I really meant by knowing the mechanism of action. The first story I'm going to talk to you about is a drug called Zelboraf, which we put on the market a year or so ago. This drug, as you'll see, is not only an effective drug for the treatment of a particular type of cancer called malignant melanoma, but it comes with a specific genetic test. And so this is a really interesting story to start with. The idea behind Zelboraf was that if you have a drug that you believe you're going to give to patients that have a disease, the drug itself will only work, we know this ahead of time, only in certain patients. But if you don't know how to select those patients, you end up giving this drug to everyone. You know that it's not going to work in 40 or 50% of them, but you don't know how to identify those non-responders ahead of time, so you give the drug to everyone. The commercial people in my industry love this, because those people who get the drug and it doesn't work end up paying exactly the same amount for the drug as the people who got the drug and it did work. Now that's probably going to change in the future when we start paying for value in the pharmaceutical industry. But for now, that's the way things work. We were paid by pill, by injection. What if you could ahead of time identify who these people were, and so only give the drug to those patients who would respond to the drug? This is what companion diagnostics, or what we call precision medicine, is doing, and it is the major force in medicine right now. We're rewriting the textbook, we meaning the universities, the academic medical centers, the pharmaceutical industry, the biotech industry, rewriting medical textbooks, so instead of being oriented towards anatomy and histology, they're going to be oriented towards molecular biology and biochemistry. If you actually take, I'm sure you guys have done this, you take Harrison's Principles of Internal Medicine, I know you guys have it in your offices, and you look at that book and you compare it to Osler's classic textbook of medicine from 150 years ago, they don't look that different. There's a section on lung disease, there's a section on heart disease. They may have called it different things, and there weren't any treatments 100 and so years ago when Osler was knocking around Johns Hopkins University. But it's fundamentally oriented the same way. 150 years later, the same textbook, with some changes, but not many. What we're trying to do now is rewrite that medical textbook so it's based on the molecular biology of the disease. So for instance, if you have melanoma, this is one of the pathways within the melanoma cell that's driving your disease. So this is a picture of a cell. There's the membrane of the cell. This is inside the cell. There's a receptor on the surface of a cell which acts like an antenna and binds to a growth factor. And that growth factor then signals through a series of different proteins, one right after the other, that that cell should divide uncontrollably. And uncontrolled cell division is cancer. So if it turns out that by chance you're walking along in the sun tomorrow, it's sunny out, and your BRAF protein gets mutated. So you have a driver up here, it's called RAF, and just by chance, in one of those dark cells on the surface of your skin, in one of those cells, the BRAF protein mutates the valine, which is the V, at the 600th position of this protein to a glutamate, which is E, V600E mutation. That happens. Then something miraculous happens to that one cell. It goes out of control. This V600E mutation now is untethered to the presence of this growth factor, and it will just continue to drive cells to divide, divide, divide. So you can imagine what happens. That one cell now has no brakes on its division. It continues to divide. All the normal BRAF-containing cells divide, and then they stop dividing when you have enough melanocytes. But the V600E one doesn't ever say no, and it grows and grows and grows until you have metastatic melanoma. What, up until now, has been a fatal disease based on one mutation, just that one. Now, hopefully your immune system took this out of play, but if it doesn't, we decided with a company called Plexicon that we would specifically build a drug that would inhibit this V600E mutation BRAF. It's called, this is the generic name for it, it's called Zelboraf. And we used basic understanding of the structure of BRAF, which came not from Genentech, but from NIH basic research about signaling pathways in cells. This was not work that we did at Genentech. This was work that was done in an NIH basic lab. We would not, by the way, we wouldn't know any of this if it hadn't been for NIH, because we didn't do this research. We were interested in actually building a drug, but the research that defined what this was, what RAS is, what BRAF is, what MEK is, what ERK is, and what are the mechanisms by which cells proliferate is all basic science funded by the NIH. But someone in the NIH did do it, someone with NIH-funded research did do it, and we capitalized on it. Actually, we meaning company Plexicon, a biotech company, all in Jackie's district, did it. We licensed it through Genentech, and we built Zelboraf as a drug that inhibits V600E. And when you do that, you take a disease that has a very poor prognosis, half the patients dying within 10 months, and this is a malignant disease, and you change things dramatically. We filed our IND with Plexicon in 2006 and rapidly saw that we could actually go from a series where the body is littered with these black melanoma spots to being almost cleared of them within 2 weeks. These are the number of patients that are responding to this drug and whose melanoma is going away. These are the numbers who are not. All of these patients are V600E patients. So in almost all of them, the drug works. This is very uncommon. Normally what would happen is you'd have about half the patients not responding. We had response rates of 70, 80% with this drug, provided that we could test for that V600E mutation. This was completed in a phase 3 study that showed that this works beautifully in these patients, and we launched the drug within 5 years of actually moving it into the clinic. Remarkably fast turnaround for a drug development and regulatory process. The reason this went so quickly is because we knew the science. We knew about BRAF, we knew about V600E, we knew exactly who to go, we knew exactly where in the protein to build the drug. All that stuff was given to us by NIH-funded research. And as a result, now patients with V600E metastatic melanoma are living many years. Many of the patients that are on that study are still alive. Whereas you can imagine if you lived only 10 months and this study was back in 2011, they would all be dead if it hadn't been for this drug. Now it turns out, I'm going to go back a few slides, that what happens when you specifically inhibit one protein is that nature then tries to work around your drug. But because you understand the mechanism of how your drug works, you can understand that too. So what we found out was roughly 6 months, actually precisely 6.7 months, after people would start taking this drug, almost all of them would relapse. what's going on? We're inhibiting the specific pathway that drives the cancer. And 7 months later, the melanoma is coming roaring back. Well, what happens is that this protein gets upregulated. So we shut this one off. Nature is clever. It says, okay, now we're going to make this one the driver of the cancer. We're going to make MEK, this next protein down in the cascade, the driver. So you can inhibit this all you want, big guy. We're taking over. And MEK takes over, and the melanoma comes back. So what do? You develop a MEK inhibitor. GSK just did that and just got approval a few weeks ago for a combination of a BRAF inhibitor and a MEK inhibitor in treating melanoma. And if you give those two together, now patients live much longer. Again, we wouldn't have understood anything about MEK and about how to deal with the resistance if it hadn't been for a basic science understanding of this pathway. But because of this now, we have a spectacular drug for this disease, we have resistance, and we understand why the resistance is there, and we're treating the resistance as well. So that's melanoma. The next story I want to tell you about is one that comes from human genetics and a company based in British Columbia called Xenon. And this is a fascinating story about how we're trying to build a new way of treating pain. So Xenon is a small company. It's got 62 employees. It's the kind of biotech company that Jackie was mentioning exists probably, 40 or 50 of them in South San Francisco right now, within a square mile of Genentech. These companies tend to start up around existing biotech companies and around medical centers because that's where they get their ideas from. We decided to deal with these guys around a group of patients that were firewalkers. There's a family that this company found in Pakistan whose only job this whole family had was walking on fire in a circus. Now, initially people thought these people are just, they can tolerate pain, that's why they walk on these embers. But it turns out when you look at them, they don't tolerate pain very well. They don't have any pain. They don't have pain. They can walk on fire, they don't feel any pain. They have a disease called congenital insensitivity to pain. And regardless of what kind of pain you give them, they feel nothing. You can put a knife through their arm. They will say, the knife's going through my arm, I feel pressure, I see the blood, I feel no pain. No pain whatsoever for their whole life. They don't live a normal lifespan because they end up hurting themselves. Though there's one story of a person putting their arm on a burner in the kitchen, and the first thing they notice is the smell of their flesh being charred by it. They don't feel the pain in their hand. They go, huh. So this family, and it is a family, had this disease that allowed them to be firewalkers. And Xenon was clever enough to go, wow, what's the genetic basis for that? If we can find what gene causes this disease, is it a disease, then maybe that would be a great analgesic. And it turns out that it is a target called NaV1.7, which is a particular type of protein that sits in the surface of nerve cells, just the nerve cells that deal with pain, by the way. And it's a channel for sodium. So when this thing works, sodium flows through the channel and the nerves signal. And when the nerves signal, it says you have pain. These patients did not have this channel. So in these nerve cells, it did not signal, and as a result, they never felt pain. So Xenon and Genentech said, wow, what if we could build a drug that you could take orally, a pill, that inhibited this channel? This would be the best analgesic in the world because these patients were otherwise normal. It would be much better than opioids, than morphine or OxyContin. It wouldn't be addictive. It wouldn't have side effects of constipation, respiratory depression, and it would be a highly specific pain molecule. Now, the work that actually found out about this channel structure that I'm showing here was not work that was done at Xenon or work that was done at Genentech. It was done in a fundamental neuroscience laboratory that was interested in ion channels and how they work in signaling of nerve cells. As basic science as basic can get. In some of these laboratories, they even stick these channels in frog cells, or they stick them in yeast cells. And I know you're thinking, God, yeast cells, what can a channel in a yeast cell tell us about human disease? And the answer is, at the time, you don't know what it can tell you about human disease. The person that discovered NaV1.7 didn't know that this was going to happen. This is the unpredictability of what is disease-related research versus basic research. You don't know. And yet we have now in our hands perhaps the best pain target that mankind has ever had. And if this drug actually does what we think it's going to do, it is going to revolutionize the way we treat surgical pain, post-operative pain, inflammatory pain, not psychic pain, so we can't deal with that, but we can deal with any type of other kind of pain with this drug. It turns out that not only is the loss of function, this is by saying if the protein isn't there, then you have a broad analgesic. It turns out there are patients that have an over-functioning NaV1.7. What would that be like, to have a pain sensor that's overactive? It turns out these people are constantly feeling pain. If they walk on a carpet, the carpet feels like it's on fire, even though it's just a carpet. So, overactive channel, a pain syndrome, inactive channel, no pain. It's the best validated target we've ever had in all of neuroscience for pain, ever. It's much better than opioids. And we're developing a drug. There are a couple of other companies that are working in this area. But again, the basic science that allows us to do this was not work that was done at the biotech company or the pharma company. Our job is to take that basic science and try to build a drug out of it. We did a deal with them specifically where we actually paid money to the Canadian biotech company in order to license the early compounds they have yet, and we're not yet in clinical trials, so this is still in this 1 in 10 kind of ratio that I was talking about earlier, in order to bring the drug in. And the reason I mention this is because every pharmaceutical company right now is highly partnered with the biotech industry and with academia. At Genentech alone, we did probably 800 partnerships last year, most of them with universities, to try to get basic science understandings to move into our company, to try to get NIH-based research to help us understand how we can use that to make drugs. They benefit because they get capital. We benefit because we get the knowledge of it working with them. We also work with biotech companies. In fact, this is what keeps the biotech sector alive, because very few of them are ever going to have marketed products. Almost all of them are going to do deals with us in order to allow us to do the development. Because it's too expensive for us to do it, it's inordinately expensive for them to do it. Now let me end with a final story about which is fundamentally a new way of treating cancer that has a manufacturing end to it. Because one thing that happens when you're building these large biologics is that it's very complex to make these. And as a result, they're almost all made in either the United States or in Western Europe. This is one part of manufacturing that cannot be outsourced to China or India right now. And these new types of drugs, I'm going to mention a drug called Kadcyla, which we just launched, are even more complex than the standard antibody. And as a result, require the ingenuity and the innovation that comes in the United States and in Europe, and it isn't yet present. As I said, if science funding continues at its pace in China right now, it will be present in China shortly. The drug is an antibody, similar to the one I showed you, but it's got bits on it. These bits are shown here in red stars. These are standard chemotherapeutic molecules, very nasty molecules that taken by themselves would be too toxic. And what we've done is we've linked them physically to the antibody. So the antibody becomes a Trojan horse. The toxic molecules, the poison if you wish, is attached to the antibody. The antibody goes into the cancer cell, releases the toxins only inside the cancer cell, and kills the cancer cell. This was the idea that we, Seattle Genetics and Immunogen had to build what we call antibody-drug conjugates, and Kadcyla was our first. These are complex to make. You have the standard antibody that you normally are used to, and then you have a whole bunch of chemistry that attaches you to this toxic drug. Now, what happens then, the first one we decided to work on was a pathway we were familiar with. In breast cancer cells, I think many of you are familiar with the protein HER2. It's a growth factor receptor. It's the drug that Herceptin binds to. Herceptin has changed the life of women with HER2-positive breast cancer. And Herceptin is an antibody that binds to this receptor and shuts it down. What we then did is simply attach the poisonous molecules to Herceptin, and that molecule is called TDM1 or Kadcyla. So think of it, Herceptin with this added kick of having a toxin attached to it. It gets internalized into the cell, and for those of you that are cell biologists, you'll recognize some of these names, but basically what happens is the cell chews it up, releases the toxin, and the toxin kills the cell. But because it's only happening inside the cell, it's much less toxic than chemotherapy. Now, it turns out that this is really effective. This is an animal model that is HER2 positive. So here's the controlled growth of the tumor over time. So if you do nothing, the tumor grows, 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 and grows. If you give it Herceptin, well, it grows, but not quite as quickly. If you give it the toxin as much as you can, which isn't very much, plus Herceptin, maybe some effect, But if you attach them covalently so the drug is only released inside the cancer cell, you wallop this thing down. So in animal models, this thing is more effective than Herceptin. We then did a phase 2 study where we looked at patients that were HER2 positive who had failed Herceptin. These are patients that had already failed the antibody part of this. And we thought, is there any benefit of giving them now this more complex drug? And it turns out that it was dramatically more effective. This is the Kaplan-Meier curve of how quickly you die with HER2 positive breast cancer given this antibody drug conjugate if you're HER2 positive or HER2 normal. So we only look for patients that have the antibody receptor on the surface of the cancer cells. And there's some long-term survivals here of patients when they're given our drug. We then did a phase 3 study where we compared solely the antibody drug conjugate versus a HER2 agent and a chemotherapeutic agent. And as you can see, you survive much better if you were given the antibody drug conjugate than if you weren't. This was true for progression-free survival. It was also true for overall survival. So these patients are still alive, 34 months out. These are patients that have metastatic breast cancer. Before this drug, they had a lifespan of about 14 months. So if you look at these studies before Kadcyla, almost all the patients that were on the study are dead now. But these patients are still alive, even if you failed Herceptin the first time. And the reason is that this is a highly effective way of actually delivering toxic drugs to the inside of a cell and not to the whole body. The drug's approved, it's on the market right now. It was part and parcel of a large business development opportunity within Genentech where we worked with a number of different companies. So think of this as just economic growth. This area is now booming. Genentech itself has, and other companies within the industry now have over 25 of these types of molecules in development. There is billions of dollars being spent just on this type of drug. And because it's a large molecule where you have to covalently and very precisely attach the toxin, we can't outsource this manufacturing to China or to India. There's nowhere over there they can make this. And we have to make it very carefully. You can imagine if you have a little bit of free toxin hanging out, that's not very good. It has to be very precisely made. And so it's being precisely made in Vacaville, California, not far from San Francisco. It's being precisely made in certain centers in Europe. These are the kinds of manufacturing plays that keep jobs and innovation on the manufacturing side in the United States and in Western Europe. Again, the ways and those chemistry mechanisms I said of attaching the toxin, knowing what toxin to use, how to attach it, that's not work that we did. That's not work that Seattle Genetics did. It's not work that Immunogen did. It's work that a basic scientist did, funded by the NIH. So this drug would not exist. And those patients that I mentioned in this graph beforehand would not be alive if it hadn't been for that NIH basic research. We now have taken Kadcyla, which we've launched. We have a number of other antibody drug conjugates that are in development. I'll say we have even tens more back here. So we're putting a lot of muscle behind this new way of treating cancer. Because it means that once we find a marker on the surface of a cancer cell, it doesn't matter what the marker does. We just build an antibody to it, attach the toxin to the antibody, and then use that as a drug. It means that the future for cancer therapy is going to be a cocktail. Maybe you have 5 of these, based on whatever the surface of your cancer cell has, so that you would get one cocktail of 5, and you may get another one. And it doesn't really matter where in the body the cancer is. It could be breast cancer, it could be in your gut, it could be in your lung. It's a molecular definition of the cancer, not an anatomic definition of the cancer. What we're interested in is, where's the HER2 positive cancer? If it's in your gut, we'll give you Kadcyla. If it's in your lung, we'll give you Kadcyla. If it's in your breast, we'll give you Kadcyla, because it's HER2 positive. The description is a molecular description, not an anatomic description. So let me end by saying, what I've been trying to tell you is the importance of NIH science for the economics of our industry. Without this kind of basic science, we do not know how drugs work. Not only can we not find the drugs, but once we find them, we're very poor at developing them. If NIH funding continues to collapse at the rate at which it's predicted to collapse, then science of the type that gave rise to these drugs will leave the United States and move to other countries, primarily China. With that, almost inevitably, will be the movement of the pharmaceutical industry to where the science is. Because these global companies are going to go where the science is. And with that will move innovation, jobs, and an intellectual climate that we now are at the world's leading country in away from this country. And this is what keeps me up at night, is the fact that without basic science funding, our industry starts to collapse. And you can just march down from there to human health, the ethics of it, et cetera, et cetera. I'm going to end with this slide. For those of you that haven't been to the Genentech campus, we have 10,000 people on campus, and I'd be happy to host you there if you want to come and visit. But what you'll see is large posters of these people up around the campus. These are real patients that take our drugs. And we put them up there to remind us that although Corporate Finance 101, when you were in business school, said that the shareholder was the most important stakeholder, it's actually not true. The most important stakeholders that we do are these patients, because these are the patients that are still alive because they take drugs like Kadcyla and Zelboraf. And so we have these posters up around campus to remind us about why we come to work. And with that, I will end and take questions. Thank you very much. Yes?
I saw when you're describing the different phases of testing and whatnot, phase 2 had a 30% or 0.3 success rate. Why is that? Is that when you take it from like animal trials to?
No, all those phases are clinical trials.
Okay.
So in building a drug, you first have to come up with a drug. So you actually come up with a specific molecule. You file a large package with the FDA called an Investigational New Drug Application, or IND. And you put in there all your animal studies, your toxicity studies, your efficacy studies, how you can manufacture it, how pure it is. You give it to the FDA, and within, usually very quickly, within a period of time, they'll review that and say, yes, you can do clinical studies. Phase 1 is all about safety. You give it to a few patients, and you see if it's safe. Phase 2 is all about effectiveness. So it's the first phase where we really ask the question, does this drug now work in the complex environment of a human, where it did work in the complex environment of a mouse before? And it turns out that that's the reason things fail. They primarily fail for efficacy reasons, because this is the first time you really test the question, does this really affect the disease in patients with the disease in humans? Other questions? Yes?
I was wondering if you could speak on how and to what extent the biotech industry, and particularly, I guess, companies that deal with biologics and recombinant DNA like Genentech, have been affected by the recent Supreme Court ruling, and what you see as sort of the legislative outlook as far as intellectual property.
Yeah, I thought someone might answer that question. So I'm going to put all sorts of caveats. You guys are used to caveats, right? I'm not an attorney. This is not the Association of Molecular Pathology versus Myriad, which is the decision you're talking about that was recently decided by the Supreme Court, came down, for those of you that don't, you guys don't know this, right? So they basically saying that DNA itself cannot be patented, but other forms like cDNA, which is a constructed form called complementary DNA, can be. Most of what we do is cDNA. And so we don't think this is going to have a major effect on our industry. But we're looking at the ruling much more carefully to really look at the impact. But our initial comments, and our CEO, Sabry, has said this, and we've said this publicly, is that we think we'll be able to continue doing what we're doing quite well. Yeah?
So Battelle just released a report on estimating the U.S. economic impact of the Human Genome Project. And I wonder to what extent this molecular understanding you're describing here is dependent upon sort of effective, affordable gene sequencing, and thus to what extent you feel that there has been a big impact of the Human Genome Project on the work you're doing.
Yeah. Let me answer that in a couple of ways. First of all, the sequencing of the human genome, which was about 10 years ago.
Can you repeat the question real quick because it was hard to hear?
Oh yes, the question was what impact has the sequencing of the human genome had on our industry, and in particular for sequencing, broad sequencing of patients. Is that correct? So what we contemplated in the example I showed you was not broad sequencing of the tumor, but sequencing just around one particular mutation. But you're correct in saying, wow, if we could now, instead of just sequencing BRAF in a melanoma cell, for a patient just take some of their tumor and sequence their whole genome, and we could do it cheaply, not only would we know about BRAF, but we know about what are the other 4 or 5, because that's probably the number of drivers there were in that patient. So you're thinking ahead in the correct way. Eventually, we're going to what we call deep sequence, tumors especially, to look for not only what are the mutations that we are interested in, but what are the other mutations that may go along with it. And this is going to require inexpensive sequencing. We're not there yet. However, the answer to your question also has a second part, which is because this human genome has been sequenced, it allows us to do experiments that give rise to these drugs much more quickly than if we didn't. And it's not so much that we need to know everything that's in the genome of a patient, but we need to know that what we know is everything. So what you see what happens is that science gets really effective when genomes get sequenced as the sequencing efficiency or the sequencing percentage gets closer to 100. 90% sequencing of the human genome didn't help us a whole lot because there was that 10% unknown that we didn't know about. Once we get to 99 and 100, things got very interesting because you could really say, that's it, it's just BREF. There's nothing else because we knew the totality of it. So that has been the real benefit, I believe, of the initial sequence of the human genome that came out around 10 years ago. I'll get to you in a second. Yeah?
I think she might have been asking also about, there are about 100 pharmaceuticals that are currently circulating because of the Human Genome Project, correct me if my numbers are off. But if those are, pharmaceuticals are currently being sold by pharmaceutical companies, and as you said, the companies are capitalizing on the basic sciences being done, being subsidized by the government, such as the NIH, at what point is the onus on the pharmaceutical companies to compensate?
Yeah. So I'm going to take that question more broadly than just genome, because I think it's very difficult to figure out whether a drug came from the genome or not. It's like saying, did your briefing you gave to your congressman, was it affected by Microsoft Word? It probably was affected by Microsoft Word, but Microsoft Word by itself wouldn't do very much. So it's become that commonly used right now. But you ask a very interesting question, which is there an onus on the pharmaceutical industry to somehow pay for basic science going on in universities? And I think it's a great question. I have a certain personal view of that, which I'm not going to out, but it's not in line with what the people at my company think. But I will say that we, in the United States, and I think even more so outside the United States, benefit dramatically from NIH-based research. And in the current situation of in-licensing innovation through Bayh-Dole and through tech transfer offices as it currently set up. I think over time, that discussion I'm hoping will come out. But I think it's a great point. Yeah?
My question kind of went in line with that. How much, you talk about a lot of the scientific knowledge that you have to base your drugs on comes from NIH research. How much of it is off-the-shelf NIH research that was done years ago that you went through and found and studied? And how much of it is investigators that are applying for new grants that you're
Almost most of it, I'd say almost all of it is new stuff. I mean, you're right, some of it does take time for us to build these drugs, so some of it is legacy research, but almost all of it is the continuing story that was started with that legacy research. It may have gone, BRAF was discovered decades ago, so I can't say that discovery is recent, but the understanding of the pathway, that's all recent. So, the other thing I will say is that if universities through basic science research create a drug candidate or create some modicum of value in their technology, we actually pay them. So UCSF and Stanford got paid handsomely for the Cohen-Boyer patents that gave rise to Genentech. There's a tech transfer office that is a shepherd of at least the value of NIH research to the university through the Bayh-Dole Act. I think your question is a more interesting one because you're really talking about what about that large abyss of knowledge that is not captured by tech transfer within universities that our industry uses, and should that put an ethical tug on us to think, wow, we have a vested interest in that continuing? And I think it's a really good point. Yeah?
So there was a recent article, an overview article in Nature magazine, and it was written by two guys in this area of pharma development at Bagley and Ellis. They were talking about how there's a lot of the clinical trials, there's not a lot of transparency, and then there's also a lack of reproducibility in most of the results. I forget what the number was, but it was as low as only 10% or something can be reproduced. And then I think one of the authors, he picked out 58 or so landmark papers and tried to reproduce the results. And his claim was that you can only reproduce only a handful of them. Now, I don't know how he did and all that, but obviously that affects, so we want solid science in, this plethora age of, I get money, I need to prove myself, so I got to immediately publish something without doing a lot of quality control. That may affect you guys as well. But then also, for you guys in terms of clinical trials, this area of reproducibility, I don't want to use reinterpreted, but reproducibility, being able, any comments on that?
Yeah, so I'm going to divide that into two. So the question about reproducibility of scientific results, I'm going to divide it into two questions. One is the reproducibility of laboratory experiments. And there's been a lot of talk recently. You may have heard There have been studies showing that only 50% of laboratory experiments can be reproduced when you do them. It's a relatively low number if you're a scientist publishing in Nature, Science, or Cell, and only half of them can be reproduced. And then the other question you're talking about really is in clinical trials, how many can be reproduced? And that's a more complex question because often clinical trials are initially not powered properly in terms of the number of patients in the trial to give an accurate result. And so there's a certain just statistical chance that you may get a different answer the second time you do that trial. The other thing I'd point out is that it has a lot to do with the genetic populations you're studying. One of the reasons we do genetically defined populations is because they are less variant than an outbred population such as would be in this room. So if you're doing clinical trials, for instance, in Finland, they have a higher success rate because Finnish people are, well, Finnish. They're almost all from one founder population. You do it in the United States where your drug has to work in an outbred population like us, it becomes a totally different story. But let me go back to the first question. We deal with this all the time. We often are trying to in-license technology where we have to believe someone else's experiment. And almost all the time we take the compound in ourselves and try to reproduce what they've done. In some cases it doesn't reproduce for trivial reasons, different experimental methods. They did theirs at 24 degrees, we do ours at 30 degrees, and we get a different answer. That's just science. But you're right, there is a lack of reproducibility in general. I'm not sure it's 50%, I think that's a little high. And as for clinical trials, the way to answer that question is to design clinical trials properly so they statistically address the question you're trying to address in a population that's as close as possible to where you want to go.
Yeah, I think that, and I forget if it was that article, I'm sorry, just as a real quick follow-up question, or maybe it was somewhere else I was reading, but someone was criticizing the process of how these clinical trials not only are designed, but just the transparency behind them, you know, how they're done, they're releasing the data, that kind of stuff. So I don't know, it was just a comment.
Yeah, we can talk offline about that. I mean, I think our goal is to be as transparent as possible.
Sure.
In the end, the beautiful thing about this industry is that in the end, once you get it into the marketplace, then you'll really find out about things. And there are a few drugs that act in the marketplace differently than when they acted in humans. The regulatory agency is very careful about trying to minimize that. It can't be zero, but trying to minimize it. Other questions? Yes?
About 18 months ago, the NIH opened a new center within the NIH called NCAT.
Yes.
Center for
Yes,
Advanced
Chrysostom.
Yeah,
Yes.
exactly. And there's been some criticism on the Hill that this isn't something that the government should be doing. That's the job of a pharmaceutical company. Can you comment on that?
Yeah, I can. A couple of things. And Chris and I have been together on panels yesterday. And I guess a couple of things. One is I think the way that NCATS was initially outed to the world was that it's going to make drugs. And I think that if you accept that, then the criticism was warranted. But if you actually look at what Chris and NCATS are trying to do, it's not make drugs. It's to investigate the science that underpins the making of drugs. So it's a science institute, part of the NIH, that is interested in the science behind things such as pharmacokinetics, small molecules binding to proteins, toxicity, etc. And it's trying to build up a basic science understanding of those things that are important for those people trying to build drugs. So it's a bit of a nuanced message. The real mission of NCATS is a spectacular mission. It's more science. I can't help but love it. I don't think that their mission is to compete with us, and we don't feel that way, that their mission is to compete with us. I don't think Chris would have gone there if that. It's stupid to think that a government-funded, and I would say minimally government-funded in comparison to the amount of money we spend, and we think about $9 billion on one end, is going to compete with the pharmaceutical company, but I don't think that was its mission. I think its mission was to do things that we wouldn't do. There's certain things, repurposing drugs was one of their projects, for instance, which is something we don't do. So I've got to be supportive of someone who's trying to do that, because I think it eventually can help patients. Yeah.
I guess this kind of goes along with that question. So you've given a very persuasive presentation on why basic research funding is important, but if you look at the trajectory of NIH funding over the years, it doesn't really correlate with productivity. And I think from hearing Chris Austin talk, one of the things NCATS is trying to do is shift the trajectory, and you talked about a couple of ways, but do you think that more of these, rather than just having the basic research there, but more active partnerships between government-funded entities and government companies are going to be more important?
A couple of things. Trying to find a correlation when the timeline is so long is very difficult. I think that people have tried to draw correlations between the level of NIH funding and the level of productivity of our industry, and it's almost impossible to do so because the time lag between when a discovery is made, what was the key discovery and who made it, when was it made, and the drug entering the market may be 15, 20, 30 years. The HER2 receptor was discovered 20 years before Herceptin made it to market. So I don't think anyone's done a really careful analysis of that. It would require a ton of work. Secondly, your question was about the relationship between the pharmaceutical industry and academia. We have rich relations with academia right now. I have a great love for academia and want it to continue to do what it does. There are people that stay in universities to do basic science research that would never move to Genentech, even though Genentech is about as academic a place as you can get. They definitely would not go to other pharmaceutical companies. Those are the people that in the past have found out the mechanisms that we're using here. And so what I'm arguing for is the continuation of basic science funding by the NIH in research institutions in the United States, the continuing and increasing of that funding as an imperative, not only from the point of view of Americans' health, but from the point of view of the economy of America and the economy of the pharmaceutical sector. Yeah?
So you've talked a lot about the importance of supporting basic science research, and so I'm sure you're aware NIH I think spends about 53% of their budget on basic science. It's been about that level for the last
It's been
20
pretty
years.
flat, yep.
Do you think that they spend too much on applied research?
Well, so we were talking about this last night over dinner. I would like us to start to lose the difference between applied and basic. I suffered from this when I was trying to go into a basic lab in my background where they said, oh, you're a physician, you can't do basic science. I said, PhD student, I'm just doing the same stuff you're doing. But there really still is a solitude between the MD and the PhD in medical centers that is more a legacy of personality than anything real. So I have trouble in understanding what's applied and what's basic. There are some applied studies that are truly applied to human disease, but is understanding the molecular biology of melanoma applied or basic? Varmus and Bishop got the Nobel Prize in the late 80s for realizing that cancer genes are just normal genes that are somehow dysregulated. Is that applied or basic? I call it science, and I'm trying to lose the discrepancy between both. So I'm not sure that I would address the issue of the ratio. Yes, sir.
To what extent do FDA regulations affect the probability of technical success for these pharmaceutical companies?
Well, as you saw the number, I think it's about 80% right now is our probability of success through the NDA process. So in the end, the FDA plays an extremely important role in whether our drugs make it to market or not. I mean, on the trivial sense, they play, given that we're in a regulated market and we can't declare victory until they say so, they play an ultimate role in the probability of success. However, as I'm sure you're aware, the role that the agency plays is a tough one. Their job is to ensure that Americans have safe and effective medicines, while at the same time ensuring that safe and effective medicines make it as quickly as possible into the marketplace to maximize the benefit. And they do so the way they're doing it. You can argue about whether they're too tight or not. I believe that the part that the pharmaceutical industry owns for that is that we have to build drugs that are really effective. That's the part we have to own. We don't want to go to the FDA with a molecule that has a 3% benefit over an existing molecule. I know it may be safe and effective, but come on, guys, that's not a great use of a limited amount of capital. I think the part that we own is really, through this basic science understanding, is building drugs like Kadcyla and Zelboraf that take the response rate from 10% to 70%, not 10% to 12%. And there have been many pharmaceutical companies in the past that have played in that sort of minimal benefit game. I'd like to see us up that game so that we're playing with significant clinical benefit. When there is significant clinical benefit, the FDA has actually been remarkably good at getting those drugs approved. I can want them to be a little faster. I think that there are times they're not as thoughtful as we'd like them to be in terms of understanding some of the new science, but we have an open dialogue with them constantly about this. And we're working very closely with them so they can understand how companion diagnostics work with this, because that's a new thing for them.
You talked about the best use of scarce capital and finding most effective use. Some of the smaller firms that we've talked to, are talking about the problem that they have phase 2. Yes, you talked about that's where you're kind of testing the efficacy of it. They're also talking about problems with capital. Is there also, that might be something that your company's not having difficulties with, but are there ways that public policy could better promote more capital going into the industry?
Yes, it's a really important point. For small biotech companies that don't have products in the market and hence don't have revenues and rely totally on the capital markets, both private and public, in order to fund their operations, they're limited by the amount of capital they can raise, not by any public policy, in my opinion, not by any public policy decision, but by the markets. The venture capitalists that are funding these companies only want to fund them up to a certain amount of funding because they've got to make a return for their limited partners. So the way in which policy can help biotech is by policy being in the best interest of patients to bring the drugs as quickly as possible to market, and allowing the companies who can do that, like us, do it. And it will trickle down. I think you've seen this recently. The IPO market for biotech companies has opened up quite nicely in the last couple of weeks, especially. And it's done so primarily because large companies like ours and large biotech companies have done quite well financially. And that's when the investors start to open up their pockets for these kinds of things. The other thing I'll say is that almost every company does a deal with a company like ours in order to help them economically. So it's very rare now for a biotech company to move a product into the clinic without having some pharmaceutical company helping them do so. And that's what my group spends most of its time doing. Other questions? Yes?
Do you see a profit opportunity in conducting that basic research? Let's just say if the NIH really had no funding at all or just ceased to exist, you think there's the profit opportunity there, profit?
For us to do, you mean?
Yeah, for you, pharmaceutical companies, anybody.
Well, I think, so here's people have asked me that question. What happens if NIH funding continues to go down, or as you point out, even in a nuclear option just goes away? I think the most likely probability then is that science will move to another country. I don't think we'll start doing it in America. I think what we'll do is that science will go to places where it is being funded, right now primarily China. And with that, eventually we would move there. So the 12,000 employees that Jackie Spear has in her district right now in South San Francisco overlooking the airport will all move to Shanghai or Beijing. Actually, I personally probably won't, but that's where we'd move. We'd move the group just in the same way that Basel moved to South San Francisco. South San Francisco will move to where the science is. Global companies will go to where they can do the science in the most cost-effective way they can. I don't think pharmaceutical companies are ever going to get in the business of basic science research in any big way because we're having trouble meeting our cost of capital now. Why would we layer on more billions? And as I said, we spend $9.5 billion in research at Roche. That's more than Novartis, that's more than Toyota, that's more than General Electric. That's the largest research expenditure of any company, period. More than Apple, more than Microsoft. And with that, we're only doing minimal amount of basic research. We cannot afford it. And we don't, as I was mentioning to someone else, we don't have the right people. The people that are really good at figuring out these pathways, they like to stay in academia. They want to follow their nose and be academic scientists. We hire as many of them as we possibly can at Genentech, and I think we're better at them than most. For instance, our former CSO just wanted to become president of Rockefeller. He's back in academia. So we do better than most companies do when it comes to hiring academics. But most academics don't want to go and work in a company. They want to be able to follow their scientific nose in a research institute, and that's where the most creative work has happened. And it's happened for the last 20, 30 years in the United States because of NIH funding. All right, if there are any questions you didn't want to ask in the crowd, I'll be up here for a while. Thank you very much for your attention.